Hello, everybody. I'd like to take this opportunity to welcome you to our 15th annual Darwin Week celebration. And yay! I am super excited because we have had a great week so far. Darwin Week was established as an international event to celebrate the impact that Charles Darwin had on the scientific community. Many people have heard of the book On the Origin of Species. However, that is just a single time point in the history of evolution. And over the years, it has continued to grow and change and become the theory that really underlies the biological sciences today. So we're really excited to not only just celebrate Darwin, but his lasting impact on science. I would like to take a couple seconds to thank the committees that helped organize Darwin Day, especially Danielle Pritchard, who is the TJC chair for Darwin Day. She's right there. And Wei-Chin Ho, who helped with the movie. He's up here. We had a fun time where we had booths on Tuesday where students could come and interact with some of the museum species and do some art. We had the movie Evolution Earth on Wednesday, right here on this screen with some popcorn and soda. On Thursday over at TJC, the students got to hang out with our speaker, and then there was a showing of Serengeti and the planetarium. And then we had our first keynote talk from our speaker. And now today we've had a great day. I'd like to thank the students who participated in the lunch with the speaker. I know they had a good time, so thank you for that as well. If you have smaller children, I will highlight we have one more event this week. Tomorrow we are at the Discovery Science Place, and a bunch of students from the university are going to be presenting some museum species. We've got some live animals. We're going to have some games and some art projects down at the Discovery Science Place. And so this is a kids' science museum. So it's probably not as many undergraduates' cup of tea, but if you have a sibling like 5 to 10 years old, might be a great chance to take them down and introduce them to some of the natural history and evolutionary science that really is a fantastic opportunity for them to learn. All right. Now, I did want to take a couple minutes to also introduce our speaker tonight. We have Dr. Michael Muehlenbein, and he is from the university, or Baylor University in Waco. I just wanted to tell you a little bit about his journey. So he received his Bachelor's of Art in Anthropology and Environmental Science from Northwestern University up in Chicago. He then got a Master's of Public Health and Tropical Medicine at Tulane University, just across the border in Louisiana. He then moved on and got a Master's in Philosophy and a PhD in Biological Anthropology from Yale University, and then started a string of faculty positions where he continued to move up, starting at the University of Wisconsin in Milwaukee, Indiana University, UT San Antonio, and finally finding his home in Baylor. He's also gotten a certificate in travel medicine. If you were at his talk last night, you heard a lot about his work in travel medicine and public health associated with that. And currently he is the professor and chair of the Baylor Anthropology Department. Some of the fun things about him: he has worked in 11 different countries. So as a biological anthropologist, you get to travel. So if that's something that really interests you, maybe you should look up biological anthropology and talk to him about his program afterwards. And what I think is a gargantuan effort, he has taken 16 different study abroad trips with over 250 students to foreign countries to do different types of research. He's been the regional director for the Waco area for COVID. and he's published over 100 papers, which is a lot of work. Finally, he's edited 2 different books. One is The Basics of Human Evolution, and a second is Human Evolutionary Biology. And so if you're really interested in kind of where we come from and how we're related to other species, those would be a couple good books that you can look up. Without further ado, I'd like you all to welcome Dr. Muehlenbein.
Thank you, Brent. Can you hear me okay? I appreciate that introduction. I should take you everywhere with me, right? No, thank you for the invitation to be here. It's a pleasure. I had a great experience. I mean, coming in not just for a lecture and leaving, but getting to hang out and meet with a lot of faculty. Felt like a job talk, but without the pressure, which is pretty cool. So in thinking about what I wanted to offer and how I identify myself as a scholar, what I do is just so diverse and to a fault. And I accept that. And it really wasn't until our experiences with COVID that I started to identify mainly as a father, and that everything else that I'm doing and that I'm going to talk to you about tonight allows me to do that. Before, I used to live to work, and things changed. It changed for all of us, right? So to keep that in mind, and I put that up there because that photo was taken 8 years ago tomorrow. So if you had to divide my work broadly into 2 different topics, it would be evolutionary medicine and One Health. One Health is what I spoke about last night, and that is the idea that humans, wildlife, domestic animals, and the environment are all interconnected. And to fix one issue in one group, problem, or area, you've got to be thinking of holistically. And that has taken me to all these different countries to look at the risk of zoonotic disease transmission and what people think about it and how they perceive these risks, because we just came out of a pandemic of zoonotic origin, but it seems like people are already forgetting, right? And so we're destined to repeat ourselves. So what I'm going to talk about here is my work in evolutionary medicine. Show of hands, who's heard this before, evolutionary medicine? Yeah, okay. So it's the basic idea that you can utilize evolutionary theory to understand better medical research and practices. There's, classically an understanding of the evolution of menopause, for example, not just the how, but the why. And in evolutionary medicine, we think about both, not necessarily the treatment or even the prevention, but why something materializes that affects our health. And arguably, in order to develop all the treatment programs that we have, you have to think broader than just how it works, but again, why. So what I do is I use life history theory primarily. This is a part of evolutionary ecology that tries to understand phenotypic evolution, basically all aspects of an organism through development, conception, all the way to death. And the idea is that, there's several, but one of the main tenets of life history theory is that there are trade-offs, okay? Trade-offs in life, you know this. Mick Jagger said, can't always get what you want, try sometimes, you get what you need. Can't have your cake and eat it too. So here the idea is that as developing organisms, and I'm going to say we, when I say we, where I'm talking about the human species, but certainly absolutely not limited to just humans. But we have to deal with competing functions, reproduction, growth, well, let me reverse that. First growth, then reproduction. But other things like maintenance and storage. And this is where the endocrine system comes in for me. So while I was doing an MPH I did one in tropical medicine, then I did another in biostats. But I'm a first gen student. My dad, electrician, mom, homemaker. So my mentality is to work, right? And so I was working at Louisiana State University's Department of Endocrinology, but also the Tulane Primate Center. And I wanted to combine these interests. And it seemed that hormones, or the endocrine system in general, was that connecting factor for me. So the idea is that chemical communicators throughout the body can facilitate these types of physiological trade-offs, right? If you have an increase in one hormone, it may have multiple effects throughout the organism. And when we then therefore test for your hormone levels and we see variation, well, there's an incredible amount of variation, but also variation below and above what is clinically considered important, we start to question whether or not we have to correct that. We have medicalized a lot of aspects of our physiology, right? The menstrual cycle is a great example of this. In this case, you have hormones that vary, and they specifically do this so as to increase or decrease the likelihood of implantation and therefore a successful eventual birth at any given time, right? Now, there are fundamental differences between male and female reproductive physiology that you're all, I'm sure, aware of. But in this case, the idea is that females, and demonstrated in a number of different species, okay, when undergoing very mild exercise regimes or dieting, okay, you don't have to be a ballerina for this to happen. But what's happening is you're getting decreases specifically in estradiol and progesterone, and that can cause things like amenorrhea, in which you're no longer cycling. This is considered a clinical manifestation that requires treatment. The life history perspective here would say that this is an evolved mechanism to better time reproductive events. That is, the endocrine system is acutely tuned to external stimuli. Okay, in this case, it comes in the form of energy flux, how much is coming in and how much is going out. And it doesn't take much to alter this particular system in females. Males also have variation in reproductive steroid levels. In this case, I'm showing testosterone across 4 different populations. And what we know is that it doesn't have that monthly cyclical pattern, but rather there seems to be, at least for a lot of males, a consistent decrease with age. Okay? People say, oh, it's 1% for every year after age 40 or something like this. Now, the ecological perspective would be that it's all relative. So if you look at the top, these are males from Harvard, Boston. If you look at the bottom, those are the Ache, which is a hunter-gatherer group in Paraguay. So it's not that they're experiencing, that we should be surprised that they're experiencing lower testosterone levels throughout life. It's that we start much higher, and that decreases with time. So the question is, why? Why would it start higher for us? Okay. Well, testosterone levels in males vary in response to a number of different factors. Aging is just one of them. Okay. It responds to competitive action. Okay. So if your team on Sunday loses, it's very likely that we could test you and see a decrease in testosterone. I'm not going to go in. It's really interesting to think about the how and the why here, but we don't have time for it. Another thing that affects average T levels in a male is partner status, specifically parenting, right? It used to be, dads have lower testosterone, but is that because they started with lower testosterone, or is it because they had a kid and it went down? And they've actually shown that it's the latter, that it and independent of age. And then finally, T levels respond to illness. Okay, and this is the important thing I want you to remember. So I've been driven by a few fundamental questions. That is, how, from the proximate perspective, from the biological, how costly is immunity? How are hormones immunomodulatory, and how do they change in relation to immune status? Okay, that's the how. And then the why is the ultimate explanation. This is Nico Tinbergen, obviously, right? So levels of analysis. Why have I been drawn to this? Well, humans have co-evolved with pathogens since before we were a species, right? And yes, before 10,000 years ago, the major sources of mortality and morbidity were probably things like predation and childbirth. But as we became sedentary following agriculture, the development and spread of it, we developed critical masses that would facilitate the spread of pathogens, okay? We have evidence of this going back at least 3,500 years in mummies. So tapeworms here in the mummies in the left. Pinworm from coprolites in Southwest United States from thousands of years ago. And this is not a problem of the past, obviously. We're thinking about things right now like monkeypox and avian influenza, right? And if you ever, for the students in the room, if you ever see some type of specific number like that, it's almost a guarantee that it's wrong, right? There's no way. Because with new genomics tools, we can diagnose many different things more efficiently than at the subspecies level and strain level and so on. But the point here is that this is still a problem. You don't have to go and travel somewhere exotic for these things to happen. They're happening here. I would say that a good estimate would be that 5 to 10% of you right now have a respiratory infection. It's just the general stats for this part, for Texas. What is your body doing, and how is that influencing other physiological systems? Okay. So in response to evolutionary pressures from pathogens, we have developed a very complex immune response. This is a terrible simplification of it from one of my papers a long time ago. But if you're taking immunology, you will realize that it is way, way more complicated than things like T and B cells, right? There are reasons for this, okay? There are compensatory mechanisms. There are trade-offs that are happening within innate immune functions at mucosal levels, and so on. Something this complicated must be energetically costly, right? The question is, how much? Because if immunocompetence is really costly, it should be involved in these trade-offs, right? We know that reproduction is costly, especially for mammals, mammalian females in particular, right? Because they're bearing the brunt of gestation, parturition, lactation, and so on. But what about the role of other systems that are competing for those resources? So I'm going to tell you about a series of studies that I did to try to answer some of these questions. I'm not going to tell you in the order in which they happened because I was misguided in my first studies, right? And hindsight is 2020. So I want to highlight a few different things. One, it has been a lot easier to quantify the energetic costs of immunocompetence in other species. You can imagine why. Well, we can infect them. It's ethically and logistically challenging for me to get permission to infect you. Doesn't mean it can't happen. And it has happened in clinical trials with rhinoviruses, for example. So for human work in biological anthropology and human evolutionary biology, we're limited to more of these natural-like experiments. So passive surveillance, waiting for people to get sick, and then seeing what's happening throughout recovery. Okay. And in one case, we recruited 25 college male students who had an upper respiratory tract infection and then looked at changes in resting metabolic rate and hormone levels and all sorts of things throughout recovery. Now, the important thing here is that this is in the absence of fever. We've known for a long time, for at least 30 years, I think the first paper was, that fever is associated with a significant increase in metabolic costs. Okay. But even in the absence of fever, your immune system is ramping up. And that is energetically costly. In this scenario, it's not that much, 18% or so increase. And that's fine for me because I've got the energy to spare. I've got all this storage, right? But this can be, if you think about the evolutionary history of this, as well as what's happening in LMICs, low and middle income countries, and including here, this might be significant. So because immunity is energetically costly, it should be involved in trade-offs with other functions. And for the most part, this has been studied under the context of growth, child growth. Right? We know that stunting can occur for a number of reasons. And it could be, just lack of available food, but there could be other ecological reasons for it. But in naturalistic studies, kids in Indonesia with whipworm, elevated C-reactive protein levels in the Chimani of Bolivia, or IgE in the Shuar of Ecuador, we've seen this. So those who have higher immunological costs because of infections are typically stunted. So it's all relative. If this happened, and I mean, Trichuris, the whipworm, it's estimated about 20% of kids in the United States will get it at some point in time. You can go to CVS or Walgreens and get medicine for this, okay? You don't have to go out in the woods. It's around us all the time. So how does this affect reproduction? Not growth in this case, but reproduction. Why think about this? Because reproduction is the rate-limiting step for evolution, specifically the uterus. Okay? But I mentioned before fundamental differences between males and females. And with females, reproductive biology, as translated through changes in these hormone levels, is very sensitive for the most part, for most people. For males, it's not. So if you put males under the same dieting and exercise regimes, testosterone levels, spermatogenesis, sperm quality or quantity are unaffected for the most part. And even if they were, okay, it doesn't take much for reproduction to still happen here. So we don't think about male reproduction in terms of spermatogenesis. We think about it in terms of attracting a mate, competing for mates, protecting families. Okay. These are things that are in fact facilitated by different hormones, testosterone in particular. Okay. This is taking studies that we've demonstrated very well for a long time in non-human animals and understanding it in the human animal. Okay. So the thing that is associated with a lot of these is anabolic functions, muscle mass. Right? You can imagine that under certain circumstances that this could facilitate. Now, I'm not talking about looking like this guy. And you don't typically see this naturally occurring. Why? Any ideas? What? Just yell it out. It's not built to withstand the costs of maintaining this. So testosterone being an anabolic steroid, it facilitates muscle anabolism, right? But at the same time, it causes things like fat catabolism and increases your resting metabolic rate. You can imagine that if you are Schwarr or Chimani, you don't want to do that. Okay? So we do see this global variation, starting at lower levels in places where you do not want to incur the costs associated with high levels of testosterone. Some of the costs, elevated energy expenditure. It takes an enormous amount of calories to obtain that and to maintain that, right? You also have increased risk of prostate cancer, free oxygen radicals, and then finally immunosuppression. It's this last part here that's actually been very questionable over the past few decades, especially as it applies to humans. There's a lot of work on this in non-human animals. Why? Because you can manipulate them more easily. Desert fence lizards, found in Texas. This blue dewlap sac that is used for attracting a mate. So you've seen this behavior out in the wild. What do? They get up and they do push-ups to attract the ladies. So if you take testosterone capsules, slow release capsules, and you put them under the skin of these animals, they do more push-ups. I mean, a lot. Okay? And they're more likely to die. Why? Well, at first people thought, well, you're doing this and a predator is going to be a hawk coming by. It's going to see you doing it. It's not because of increased predation. It's because of increased metabolic costs. And you see this in other species too during the breeding seasons. Deer, male deer will forego eating. Red deer in Europe in particular, they'll stop eating because all they're doing is concentrating on reproduction. So how are these things? It's costly. We know that. It's facilitated by hormones. We know that. How are these immunomodulatory? And I'm very intentionally using the word immunomodulatory because for the longest time, people have said that testosterone is immunosuppressive. It decreases your immune response. Well, I mean, immunity is enormously complex, okay? What aspects of it are being affected? Because it's not like it's just a blanket response. So we know that variation of immunocompetence is under control and that we would expect to see variation in hormones that they co-vary with things like nutritional status, presence of a mate, disease risk. So environmental signals, much like the environmental signals I was talking about before for females. So if you do a systematic review and meta-analysis on this in humans, okay, we see that it does appear to have specific effects on T cells, inhibiting cytokine production, antibody production, natural killer cells, macrophage activity. But honestly, the results are all over the board. You find one paper that says yes, you find another paper that says no. And when you see something like that, that's a signal to me that variation is natural. Okay? We tend to categorize things, specifically human medicine, right? We want to say sick or not sick. And if you ever go into a CBC, you get the cutoff values, right? Those are not necessarily updated. They're certainly not tailored to you. Okay. So I want to reconcile mixed results, especially in vivo studies. If you do some in vitro, stuff, or ex vivo, it's much more consistent. But in vivo, in humans and non-human primates. Why? Because I'm a biological anthropologist. It's just the species that I chose. Okay. The difference really just the model system. So going back way before that first study that I mentioned, I started working with wild chimpanzees. Southwestern Uganda, there's a forest known as Kibale, and there are several habituated groups of chimpanzees. In Gombe, Tanzania, I'm sure you've heard of Jane Goodall's group, right? Jane Goodall's group was about 18 animals. I was working with the Ngogo group, which is about 300 males alone, okay? Fully habituated. This is a fission-fusion species. 300 never come together at once. They're in groups, coming and going. But the good thing is they're habituated, they're identified individually, and so you just integrate yourself into this community. It was hard, sleeping in a tent for a year and following them around, picking up their poop. That was my job. Yeah. Again, my first master's was in tropical medicine, which is essentially parasitology. So we're doing basic parasitological analyses, but also we developed some assays for measuring steroid levels in fecal samples. Not easy to do. Easier now, that's for sure. But at the time, especially, I don't have electricity. I don't have running water. So I have to dehydrate the samples. And that's a way different type of assay too. Can you imagine? I'm not baking cookies in that. Right? So what did we find? I found that, yeah, both testosterone and cortisol positively associated with parasite richness. Now, richness, okay? I don't do egg counts like some people do because egg counts vary tremendously. And they're really a function of the fecundability of the female worm, okay? Instead, we prefer to use parasite richness, which is the number of different species that the host is infected with at any given time. Okay, because the more different species, and this is this is the norm in nature, right? When you're going out and finding a healthy animal system to study, you can expect that they have developed a degree of tolerance with these pathogens that they've co-evolved with. But intestinal infections in wild animals typically have a very skewed distribution, where a few animals will be heavily parasitized. Most will just be tolerating low-level infection. Okay, so yeah, those who are really bad off typically, in this case, had higher T and cortisol, and that would suggest that testosterone may be immunosuppressive. Certainly the dominant males had the highest T. We know that. and they were also the worst off in terms of their health. So this is why dominant male chimps, they don't hold tenure forever in the group. It's only a few years before they get kicked out. But the thing here is we don't know the directionality of these associations, right? Is it the hormone that is making that animal more susceptible to a worse infection? Or is it the infection that is causing change in the hormone? We don't know. So I needed a better controlled system to look at these things, and started working with USAMR, United States Army Medical Research Institute for Infectious Diseases in Fort Detrick, Maryland. This was the Army's facility that was originally developed for biological weapons research. And then when the treaty happened back in the 80s, it started to be used for diagnostic purposes. So rapid tests for things that might become used in bioterrorism. So I jumped on a project of captive macaques here because controlling for previous exposure and controlling for infection dose. Can't do that under natural systems with some of this, with primates especially, or humans. Okay, so in this case, we're infecting them with something novel, Venezuelan equine encephalitis, why it was considered by the Army at the time to be of great importance. So we look at changes in, so what predicts recovery? Okay, so in this case, we can sample before, during, and after, which you can't do under most natural circumstances. I'm sorry. And what do we find that in this case, now I'm getting closer towards understanding directionality. That T levels start out high. Those animals that started out high have really suppressed cytokines. And throughout recovery, they have a significant decrease in testosterone. So it's not the baseline level that we're, should be thinking about, but rather it's the change that is happening. Well, I wanted to do this work with humans. Again, very challenging. Had to find a population that is naturally infected with something that can generate a big enough response. My advisor at the time was working with malaria, so I identified a small hospital in rural, it's called Tocoa, which is a city in Honduras, about 2 hours away from the coast and equidistant from the country's northernmost borders with Guatemala and Nicaragua. A highly malarious area, usually. Of course, when I went there, there's a huge dry spell, and you need water for mosquitoes to breed and then transmit malaria. And so this is a downside of passive surveillance. You're sitting and waiting. It takes a long time and it's very expensive. So eventually got enough for statistical power to show that to again sample during infection and then throughout recovery. This is problematic. I absolutely admit. I can't sample them beforehand. That would be preferred. But that would require me sampling the entire population and waiting for someone to get sick. Can't afford that. Also, this is most cases where people live in areas that are endemic for malaria, they've had repeated exposures in the past, right? So this is going to affect their adaptive immune responses. So in this case, again, T levels are directly correlating with parasitemia, okay, number of infected red blood cells at any given time. And that T levels were lowest when parasitemia was highest. Again, now I'm really starting to think about directionality. What caused that to happen? And I don't want to get into the detail of how, but we've demonstrated some endocrinological cascades through the HPA, hypothalamic pituitary adrenal axis, and changes in LH levels in particular, or GnRH levels, that seem to be associated with this effect. So if you look in the literature, well, it turns out that T levels are really low in animals, including humans, that are infected with a variety of different pathogens. Okay? In this case, the literature is full of it. And people, if you're thinking about it clinically, what caused what? Are we just getting a bunch of people with really low hormone levels that are sick? Did that predict their likelihood of getting sick? Or did being ill result in this change? And why do I ask this in particular is because hormone supplementation for a number of conditions is standard clinical procedure. For example, those who have HIV. You think why? Well, why is to increase mood, to increase mobility, to get people off the sofa. Yeah. Okay. But we got to think, is this a system that is broke and requires fixing, or is this our natural response? So how do these levels change in relation to immune status, and then why? The idea here is that changes in hormone, specifically decreases in anabolic steroids, not just testosterone, could prevent immunosuppression and redirect energetic resources away from anabolic functions that you should not be doing at that time. Thinking about laying in bed sick with flu. You are catabolizing tissues. I mean, my wife, whenever she gets sick, she uses it as an excuse to diet, right? Because, typical sickness behavior is anhedonia and decreased appetite and other things. Whereas the opposite, during times when you're healthy, you see elevated levels that might increase investment in anabolic functions, and that we use that as a proxy for reproduction in human males, at least. So if you look at the amount of energy that goes in, you have what's coming out. Spermatogenesis, again, is a very small investment, less than 1% of basal metabolic rate in humans. I'll tell you, it's way different in things like Drosophila, but we're not going to go there. So then the body has a choice, because it cannot invest equally between things like reproduction and survivability or maintenance. And it is hormones, in this case testosterone, but testosterone does not operate in a vacuum. There are a lot of other things that are going on. So using natural experiments in humans in the field is a completely different animal than doing laboratory-based work. Because, again, limitations on study design. And of course, those limitations translate into decreased confidence in things like causality. But human biology had been really overrun by a few field-friendly techniques. Mainly things like measuring salivary IgA, which is at the mucosal, on mucosal surfaces, Epstein-Barr virus levels, C-reactive protein, and a few other things. And the literature became filled with these as markers of immunity. And that's problematic, because they represent only a tiny fraction of what is happening. And then other things, CRP took over the literature in human evolutionary biology, but we know that CRP varies by cardiovascular fitness levels. Okay, so in other words, my CRP levels are up right now because I'm stressed. not in a bad way. It's just stimuli. So one of the things I've got a PhD student defending on Friday, February 28th. And part of his dissertation included the development of a bacterial killing assay. It's so simple, it's ridiculous. Okay. It was originally used in those studying ecological immunology in birds and a few other species, where you take a small blood sample, you incubate it with different bacteria, and you see what happens. So we developed it for use originally in blood and in different species, but also saliva. And you can think that's a little bit complicated because your saliva is full of bacteria that can be grown on an agar plate. So it depends on what you're looking at. We eventually chose a very basic E. coli, not 0157, but a standard strain. And we are able to, with the proper dilution and modulation, removal of heat shock proteins, essentially, we're able to use this as more of a functional measure, because it represents what may be happening in your mouth at any given time. Of course, there are problems with dental hygiene and whatnot, but I don't want to go into those details because that's for my grad student. But we employed this method under a number of different circumstances. If you look at healthy individuals, you see positive relationships between testosterone and killing. But didn't I just say testosterone is immunosuppressive? So in evolutionary ecology, when you expect a trade-off and you don't see one, we typically call that a phenotypic correlation. And that's where this organism, this host system, has enough resources where it doesn't have to have that trade-off. I call it the Bill Gates effect. Bill Gates can have a nice house and a Ferrari. In my lifetime, I might be able to afford one because I'm resource limited. He's not. So in healthy individuals that aren't resource limited, we don't see these trade-offs, so we shouldn't be surprised by this. Okay, so what do we know? Labeling testosterone as globally immunosuppressive is obviously inappropriate. It depends on the health and energy status of the host. What about age? So infecting humans, although it happens at some frequency, is difficult and expensive. But giving humans hormones is very common, right? I mean, there's some things, obviously some garbage that's available over the counter that really doesn't work, but prescription wise. So what we did here was we recruited some aging males, and we shut down their HPA axis. So they could no longer naturally produce testosterone with a GnRH agonist. And then we had groups where we replaced them at different levels. Low levels all the way up to what we would call supraphysiological. I mean, 600 milligrams per kilogram per week is like enough to grow antlers. This is a huge amount. We're supposed to do this for a year. We had money on the grant for this, but we actually stopped at 5 months. Can you guess why? I love it. That's the first time anybody's ever said that. No, actually, hematocrit level. So anabolic steroids, not just testosterone, but testosterone in particular is associated with hemopoiesis. So especially the development of red blood cells. You can think about it because that bigger body needs more hemoglobin, more oxygen, right? Well, the problem is if your hematocrit, which is the density of those cells, gets too high, you're basically turning into a solid, right? And so we had to stop for clinical reasons. But if you look at the graph, the figure. Those which had the highest dosage had the highest white blood cell count. Actually went up. Again, these are healthy guys in which we're just controlling testosterone levels now. So general white blood cells, that's all of them grouped together. It's a terrible measure, I know, but it's basically all PBMCs. If you look specifically at different types of white blood cells, you do get decrease in lymphocytes. And this has been demonstrated in other model species. So we find it in humans. What does this mean? Well, my news feed, maybe it's because Google knows my age and my gender, but I'm getting stuff about hair loss, even though I'm not having that problem yet, or erectile dysfunction, or testosterone supplements. Deodorants, so hand gels, injectables, obviously, and it is not just targeted towards populations for sport, competitive, or aesthetic reasons. It's almost forced on us now that we should be monitoring this, because again, people get on that bandwagon that T levels are going down with age. They're not really going down enough for most people to actually have an effect. It does in some. And yeah, and it can be useful under a number of circumstances. So the thought here is that altering hormone levels responsible for these trade-offs might actually do more harm than good. And there are classic examples of this in females, right? The idea was that, I'm actually jumping ahead to a different thought and slide, but the idea has been that in order to normalize menstruation to 31 days-ish, that for a long time, for decades, the norm was to provide hormone supplementation, right? Combination of estrogens and progestins. And then you also saw this in menopausal women. The idea was that, well, if they've got that big drop in estradiol, let's bring it back up to increase libido and other things. Well, it turns out that doesn't work as I just described. We thought, oh, it might be cardioprotective or decrease your likelihood of stroke, and it actually does the opposite. So I worked as the epidemiologist for Baylor and for McLennan County during the pandemic, and had the opportunity to recruit some individuals. But ancillary to that, we wanted to look at the time, it was suggested that androgen should be used as a treatment for COVID. Okay. Why? Increase energy. And it's been demonstrated that T levels tend to be low in men who progress to severe or fatal disease. Okay. But again, what direction is that causality? So this study, we took 500 families, and this started early in 2020. Okay, the reason why was we got a few grants to look at the likelihood of asymptomatic transmission. You probably remember early on in the pandemic, people said, "Oh, you can only transmit it if you're symptomatic." Right? Well, that was, I don't know a virus that does this. Okay. So it now turns out that we know in retrospect that about half cases of COVID were at the time at least from asymptomatic individuals. But we didn't know that at this time. So we recruited 500 families to do repeated sampling throughout a year to see if they had been exposed, we could test their hormone levels. I'm sorry, the antibody titers. Okay. And at the time, there were some experimental, not FDA approved tests that we could use because of emergency orders. And I was able to process that in my lab because all I do is look at antibodies. So in 4 waves, you know, with a normal anticipated amount of attrition, we of the 500, 142, I see these are asymptomatic, healthy individuals with varying clinical risk scores. Remember all the conversation on comorbidities? If you had heart disease, if you were diabetic, if you were immunosuppressive, if you were overweight, all of these things. Well, what we did was a quick meta-analysis of what was coming out in the literature and developed a mathematical model that predicted likelihood of illness and then death if you do get infected. Okay. And remember what individuals were at the highest risk. These were usually obese males, people of color. Okay. So what we showed was that higher clinical risk scores were associated with lower testosterone, but only at younger age. Why not in older guys? Well, it may be because they have lower testosterone levels to begin with. Why? Everybody said age. No, not age. It's because of those comorbidities. So, you know, we're masking the real effect that's happening here because these guys were worse off to begin with. So I wanted to look at that in more detail. We went to the NHANES. This run by the NIH, a nationally representative cross-sectional sample. There are different waves that started about in 1984. No, it began earlier than that. But data on mortality, cause of death, different immune factors, and importantly, they were collecting data on testosterone. And a nice sample size, 2,500 individuals. And we looked at things like different types of mortality. Unfortunately, the data set is not that precise, only includes a few categories for death. But it did had T and some other things that we know that co-vary with testosterone. And yeah, lower testosterone levels were associated with higher mortality. And you especially see that effect. Michael, sorry, I yell at myself a lot. You see the effect here, widening. We saw this in heart disease. We saw this especially, look at the widening gap in respiratory infection. Older males here, okay, are much worse off and they got much lower T. Importantly, we don't see it in things like unintentional injuries and accidents. We shouldn't see that. So people are thinking that, well, low testosterone is something that needs to be treated, okay, because it decreased with age. But it may be that that's more of a symptom of other conditions. And as such, it may not be something that we want to think about supplementing. And importantly, testosterone is not floating around in your body in a vacuum separated from other hormones. There are a lot of factors in play here. Okay, and so we began looking at other things like DHEA, dehydroepiandrosterone. This is available over the counter. It was touted as a potential weight loss drug probably about 20 years ago. And it is a steroid, but it seems to do the opposite effect of testosterone. Okay. If you look at it during acute illness, this is a separate study. Concentrations are higher. So the ratio of DHEA to testosterone changes throughout recovery. You want to decrease your testosterone, increase your DHEA levels, and then have it normalize out after recovery. Because one thing I didn't mention so far is that even in mild cases of illness in human males, you can get hypogonadic responses. So it can get at the level that would be considered prepubertal, but it bounces back really quick, usually. I mentioned estrogens. Okay, very common still, less common than it was, a few decades ago, and is very useful for treating a number of conditions, especially osteoporosis. Okay, but it turns out that it can be too much of a good thing. It's generally considered immunosupportive by increasing a number of different factors, but there are trade-offs. In this case, estradiol, in particular, and this is the other part of that grad student's dissertation, is using that bacterial killing assay to look at some of these things. Significantly associated with autoimmune diseases, rheumatoid arthritis, systematic lupus, and other things. Too much of a good thing, right? Trade-offs. So to start to tie things together, because I realize I'm over time, we've got evidence that hormones influence immune function. We know this because there are estradiol and progesterone receptors in a number of cells throughout the body, especially immune cells. That looking at a single measure of immunity is not the direction that you want to go. You want to think about it more as that holistic system. It's energetically expensive, and that these responses are normal. It's normal. And if it's normal and a part of human variation, the evolved phenotype that we are now, we need to think about the ramifications of supplementation or changing these levels. When it comes to anabolic steroids, testosterone in particular, we need to rethink the model that lower T needs to be treated because it was the result of something, okay? It may just be confounded by the presence of all of these other comorbidities. And I mean, honestly, the reason why I've done this work to date is that I'm fascinated in documenting phenotypic plasticity in the human species and related non-human primates. Because again, this is something that has been done frequently in other species, relatively difficult for us to do it. And, in the field of ecological immunology, we want to know why there's variation between species, but not just between species, but within species, between individuals, and across time. On that note, I thank you for listening to me tonight. I'm happy to take questions. You can yell them out. Yeah.
What would you say?
I would say ask your healthcare provider. Yeah, I do not give clinical advice because I'm not a physician.
What would you be concerned about?
I would, are you talking about DHEA specifically? Yeah, yeah. Um, over-the-counter DHEA supplementation doesn't seem to do anything for the most part. Now, physician prescribed supplementation in pure forms, because you can't sell hormones for the most part over the counter. You sell what they call precursors, and that's a bunch of BS, okay? Because it's digested differently than injectables. So I would say talk to, if you really wanted to, talk to an endocrinologist and get the real stuff. I do not personally believe in all of these, what do they call it? Some of the stuff you see in your Reddit feeds and whatnot. I used to stay at the gas station. You know, those little bottles up there. Mm-mm. I wouldn't do that. But under the circumstance that you described, she's got to see a doctor.
I'm so sorry. I did not think it was gonna be that loud.
That's okay.
Okay, I apologize if you didn't mention it earlier during your presentation, but when it came to your Venezuelan studies of malaria, were your patients being treated for the disease as you guys tested it? If so, did that skew the results in
Uh-huh.
any way?
Yeah, absolutely. We're ethically required. You can't withhold treatment, which is another reason why I wanted to do more controlled studies and went to the macaques, right? Because I mentioned that previous exposure, and we can't control for that. We don't know how many times. There's no way. Okay. And then, yeah, you can't withhold treatment under most circumstances, especially when it comes to malaria. If there's a standard of treatment, it's followed. Huge confounder. I love your t-shirt. She's got Frog and Toad, which is the book series that my 8-year-old is reading right now.
So as you know, as society, we're kind of becoming more dependent, or at least there's a high dependence in antibiotics. Have you guys seen, or is there any theory that might result in impacting the evolutionary of these testosterone levels or any other hormones that you guys are currently studying?
Okay, so the role of antimicrobial agents, antibiotics in particular, is it going to affect our evolution? Yeah, eventually, but not in the time scale that we're interested in observing these things. Because would there be a heritable response in the level of variation in the hormones and response as a result specifically of antimicrobials? I mean, it takes too many generations for that to happen. So not in humans, we're not going to see it anytime soon. But taking antibiotics does change your metabolism. That's for sure. The majority of antibiotics in the United States are used in agriculture, animals in particular, cows, chickens, and pigs. Why do you give that to these species? At least two reasons. One, the conditions in which they're being raised, confined feeding operations, for example. But the other thing is that you're modifying trade-offs. You're preventing them for needing to invest metabolically in immunity. And so subsequently, animals that receive antibiotics, they grow faster and bigger. So I wouldn't recommend doing that in humans. I'm not going to comment on the agricultural industry, but this is the reason why people want that antibiotic-free meat, right? Another good confounder.
I'm wondering if you could comment on as I think about lowering testosterone during illness, is it to decrease the mating drive of these? Have people looked at the behaviors of?
In other species,
Yeah.
it is I mean, sickness behaviors in general, decreased libido, anhedonism is extremely common. I mean, why would you want to reproduce during a time that the body is getting these ecological signals of it's not a good time to do this? Now, is it specifically for that reason? I don't think at any kind of level of detail we could determine the exact one, or is it just a response? Either way, it's those who behave that way, including in other species, have higher evolutionary fitness. Yeah.
I have a question. So you mentioned earlier the emergency use authorization of COVID tests. My question is, with COVID, we saw that there was a impact on the immune system, such as inflammation of the heart and pancreas. How do you see the effects this will have on the future generations regarding hormone? I guess I would say how it's sustained.
Okay. Well, first, unless these effects are translated somehow through the gametes, this is not going to be a heritable effect, right? So, but continued treatment, because COVID's not going away anytime soon. It will circulate endemically in the human population worldwide forever, just like other respiratory pathogens do. Anticipating potential changes in hormone levels in response to illness should not, in my opinion, be a reason for withholding treatment for anything. Okay? I mean, classic example of the development of antimicrobial resistance. You know, you take a kid to, I have 4 kids, ages 8, 10, 12, and 15. At any given time, we have a 25% chance that one of us is sick. I did the math. And you know, you take them in for an ear infection. Inner ear infections are usually viral. So giving an antibiotic is questionable. And, you know, a lot of physicians won't, and I respect that a lot. Now it's debatable because taking antibiotics could at least alleviate some of the symptoms. And we are interested in immediate gratification, not long-term effects, and ameliorating pain. So would I give my kid? You know, I'd have to think about that carefully. But now I know from another friend of mine that that affects the hairs in the ears responsible for hearing. So treatment to relieve symptoms is always going to be the first action, I think. But treating the symptom doesn't treat the problem, right? I do not believe in, if there is a treatment available, I do not believe in you know, staying the course and letting your body figure it out. No. Good question.
All right, thank you.
All right, so it is, oh, do we have one more? One more question and then we'll call it because it's 7:15. So I do want to get you guys out of here.
No, I was just wondering about the use of like testosterone for people using it for gender affirmation or transitioning. Is there like noted immunosuppression within that, like after
That's a
taking-
great
or
question.
like or just long-term
Yep.
use of
The answer is we don't know. And there are people that are studying this. I know at least 2 people that are working on this, but the sample sizes are too small right now. You know, a few dozen individuals. And here's the issue. There are different types of androgen supplementation. So, and different lengths of treatment. And normally, I mean, from a scientific method perspective, it would be best to have baseline before and then throughout the transition. And that's the hard part that we are lacking. You know, you identify individuals who are either transitioned or in the process, and you don't have that baseline for comparison. Okay, because it doesn't mean that somebody with lower white blood cell count than someone else doesn't mean they have a worse off immune response. That could be their baseline level for sure. So yeah, we're missing that baseline stuff. And then you get people in way different stages of transition and taking very, very different things. That is going to require bigger studies. Absolutely should be funded.
Okay, so thank you very much all for coming. Let's again thank our speaker, Dr. Muehlenbein. I'm sure if you would like to come up and talk to him, he'd be happy to answer individual questions. We are going to have the sign-up sheets out on the tables outside on your way out. With that, have a great end of Darwin week.